Good morning, dear learners, educators, and seekers of truth, and welcome to Learning Academy Foundation from Mr. Andrea. Today, we embark on our journey through the intricate tapestry of genetic inheritance, a story woven into every cell of our bodies, echoing the wisdom of creation and the beauty of biological design. Let's begin with the key terms that form the language of genetics. What are genes? Gene is an allele that codes for a particular trait. Genes are segments of DNA located on chromosomes. Genes exist in alternative forms called alleles. So they are segments of DNA that code for traits, codes for a protein, and they're found on a chromosome. Genes determine all traits for all organisms. Genetic code is common for all living beings, and the sequence of bases determines this code. Alleles are a different version of a gene, different molecular forms of a gene. They arise by mutation. Dominant allele masks a recessive allele that is paired with it. Then genotype refers to the genetic makeup of a specific set of genes. Indeed, while phenotype is the observable trait, the physical characteristics of organisms, like for example, wrinkled or round seeds. Dominant alleles make recessive ones in expression. Mask recessive ones in expression. Homozygous means two identical alleles. When an individual has two of the same alleles, both dominant or both recessive. Heterozygous, indeed, two different ones. When an individual has one dominant and one recessive allele. Okay, so write in the comments which one are the genotypes that will show a dominant trait. Write in the comments below, if you please. These terms help us understand how traits pass from one generation to the next. Family traits are traits seen among family members. Many of them are inherited traits that are passed down from one generation to the next within a family. To explore inheritance, scientists often carry out genetic crosses. In the monohybrid inheritance, for example, there are the first generation, family generation, and then the second family generation, which one of these can be similar to the ancestral generation. So a monohybrid crosses examines one trait, say for example flower color in pea plants. The allele capital letter P is blue, listed for dominant, and lowercase letter p is white for recessive. Genotype, if it is both capital letter BB, or if it is heterozygous, so means one dominant, one capital letter, and one lowercase, it is blue flower. If it is both recessive, both lowercase, letter B is white flower. So if we see in this square that if the blue flower has both is homozygous dominant and the white flower is homozygous recessive, and then combined, we have four heterozygous flower, blue flowers. Blue because dominant is the blue one. Using probability, direct proportion, and simple ratio, we predict outcomes. In the first generation cross, so let's say it's the one that we've seen before. So one homozygous dominant and one homozygous recessive, if they combine, the second generation will be 100% of offspring dominant, but heterozygous, right? In the second generation, if we combine two heterozygous dominant, so capital letter A, lowercase letter A, if they combine both of them, in the third generation, we will have 75% of dominant can be 25% will be homozygous dominant and 50% will be heterozygous and 25% homozygous recessive. So if two heterozygous parents cross, the offspring have a 75% chance of showing the dominant trait and a 25% chance of showing the recessive one. And this is expressed as a 3-to-1 ratio in phenotype or 1-to-2-to-1. So 25% will be purple or blue in the previous example, homozygous dominant. 50% will be purple heterozygous, and 25% will be homozygous recessive, white flower. How to construct Punnett squares? You have to write the possible alleles and the probabilities of one parent across the top of the Punnett square and the possible alleles and the probabilities along the side of the Punnett square. So, if the alleles one is low uppercase dominant R and the other one is lowercase recessive R, they combine with homozygous recessive, so both alleles are, what we'll have will be 50% heterozygous and showing the dominant trait, and 50% homozygous recessive. The Punnett is a simple yet powerful tool. You have to write the list of each parent on the top and side, and then fill in the boxes by combining alleles and analyze the results to determine genotype and phenotype ratios. For each characteristic, an organism inherits two alleles, one from each parent. Homozygous means an organism that has two identical alleles for a gene, can be homozygous dominant if it's both dominant, or homozygous recessive, both recessive. And heterozygous is an organism that has two different alleles for a gene. So let's say that we have a mother, this 40 gene carrier, so she has one dominant and one recessive in the X alleles. And combined with an feather that is non-carrier, so it's homozygous dominant in the alleles of the sperm. What will be the offspring? 2 out of 4 chance of 50% will be non-carriers with homozygous dominant, and 2 out of 4 chance 50% will be heterozygous. Okay. So remind once again what is an allele. An allele is a variant of a gene controlling the same trait. Alleles usually occur in pairs, and depending on how they are expressed, they may be dominant or recessive. For example, red flower has both is homozygous dominant. If we combine with a white flower that is homozygous recessive, we will have a red heterozygous flower, red flower. Okay. Another example can be crossing two heterozygous, right? What will form will be one homozygous dominant, two heterozygous, and one homozygous recessive. The genotype is the genetic makeup of an organism that determines or contributes to its phenotype. If the parents are one is homozygous dominant and one is homozygous recessive, we will the offspring will be result in four heterozygous, so showing the dominant traits. What is determined by sex chromosomes? Biological sex, we know that is determined by chromosomes, right? So sex chromosomes do not carry an identical complement of genetic information. The X chromosome, being larger, carries many more genes than does the Y. Traits controlled by genes found only on the X chromosome are said to be sex-linked. Recessive sex-linked traits, such as hemophilia and red-green color blindness, occur far more frequently in men than in women. This is because the male who inherits the recessive allele on his X chromosome has no allele on his Y chromosomes to counteract its effect. So, the 23rd chromosome is either an X chromosome or a Y chromosome. The females carry XX chromosomes, so carry gametes with only X chromosomes, while the males carry XY chromosomes, so they carry gametes with either X or Y chromosomes. So XX chromosome means it is female, so female. XY chromosome is for male. During fertilization, the mother contributes an X and the father contributes either an X or Y. The genetic diagram shows that there is 50% chance of having a boy or a girl. Some traits carry deeper implications. We will look at 3 inherited disorders. If the father is affected while the mother is unaffected, the offspring will be, so the children affected will be 50%, while the unaffected will be 50%. The same as boy and girl percentage. The cystic fibrosis is caused by a recessive allele. Both parents must carry it for a child to be affected. For example, if you have a usual gene and an altered gene like your partner, we will have a 25% of child that has condition with altered genes only. So 1 in 4 chance. Actually, 4 chance, so 50% of carrier children that have usual gene and altered genes, the same as the parents, each parent. And 25% the child is not affected, so 1 in 4 chance that it has only usual genes. So we can say 25% is saved. So there will be 1 normal, 2 carriers, and 1 affected. Polydactyly means extra fingers or toes. Can be preaxial, radial. For example, if we have a person has 2 thumbs, or central if the extra finger is in the middle, or postaxial if it has an extra small finger, pinky finger. This one is caused by a dominant allele. So only one parent needs to pass it on. As we have seen before, if the father is carrier, both of them are carrier, we will have 25% healthy, 50% carrier, and 25% sick. Different methods. Gene therapy offers hope but faces ethical and technical challenges. Genetic tests can identify carriers or affect individuals, helping family make informed decisions. While powerful, genetic testing has limitations. Powerful, why? Because the DNA testing can be used for prenatal testing, pharmacogenetic testing, diagnostic genetic testing, contamination detection, pre-implementation genetic testing, DNA fingerprinting, predictive genetic testing, and newborn screening. What are the limitations? What is false positive? So the limitations they can happen false positive, so may suggest a condition that isn't present, or false negatives that may miss a real issue. Okay, or the true results can be true positives and true negatives. Gene therapy like CART, AAV, and CRISPR offers hope but faces ethical and technical challenges. Different methods like amniocentesis or blood tests carry risk and uncertainties. So we must approach these tools with both scientific rigor and compassionate care. We must be kind, stay caring, and compassion is everything. The next analysis is called pedigree analysis. Pedigree analysis is a process of examining the patterns of inheritance of genetic traits or disease in families. It involves the construction on a family tree called a pedigree, which traces the occurrence and distribution of particular trait or disease among family members over multiple generations. Pedigree charts help us trace traits through generations. Circles represent females, squares represent males. Generations are represented by Roman numerals on the left side of the pedigree. Death is presented by a slash through the symbol. Shaded shapes show affected individuals. By analyzing pattern, we can predict inheritance and identify carriers. Inheritance patterns can be autosomal dominant, where an affected female is combined with an affected male, and the offspring will be an affected female, 25% affected male, 25% affected female, and 25% unaffected female. Autosomal recessive is the one we have seen before, where when the parents are carriers, so both mother and father are carriers, and the offspring will be um, unaffected female, carrier male, affected female, and carrier female. So the only one that will not be unaffected is 25%. And then X-linked dominant. This is two generations. If an affected female and affected male, the first generation will be an affected male. If it combines with a carrier female, the offspring will be two affected males, one unaffected female, and one carrier female. And then we have Y-linked dominant. The same as before, we have an affected female and affected male, and the born one will be affected male. We will join with an affected female. The offspring will be 50% affected male and 50% unaffected female, because this one is Y-linked. Okay. So this is especially useful in understanding recessive disorders and planning for future generations. Let's talk about Mendel's legacy. All of this began with Gregor Mendel, a Humboldt monk who studied pre-plants in 1860s. He discovered the fundamental principles of genetics by breeding garden peas. These genetic diagrams reveal patterns of inheritance that still guide us today. To Mendel's discovery is his detailed record keeping and attention to the control experiment. What was his model species was garden peas. What did the species work so well? Why he chose garden peas to do his experiment? Because they easily grow and they have set traits, characteristics, can be height, uh, seed shape or seed color or seed coat color, pod shape, pod color, or flower position. Okay, and there is easily to observe. Character differences mostly in choose. For example, for the height, the dominant is the tall, the recessive is short. For seed shape, dominant is round, recessive is wrinkled. For seed color, dominant is yellow, recessive is green. For seed coat color, dominant is green, recessive is white. For pod shape, dominant is inflated, while recessive is not inflated. Colors, pod color, dominant is green, recessive is yellow. And flower position, dominant is axial, recessive is terminal. So Mendel's work reminds us that faith and science are not opposites. They are partners in discovery. His discipline, patience, and curiosity reflect the divine order in creation. As Psalm 139 declares, I praise you because I am fearfully and wonderfully made. Genetics reveals the fingerprints of God in every cell. Each trait, each chromosome, each inherited gift is a testament to divine craftsmanship. Let us teach with reverence, learn with humility, and serve with love. If this message inspired you, please like, comment, share, and subscribe to the Learning Academy Foundation. Together, we build a community where science meets spirit and education becomes transformation. Thank you for watching, and may your learning be fruitful and your hearts be full. See you on the next video. Bye.